For decades, oncology relied on a geographic model. Treatment protocols were dictated by where a tumor was found, whether in the lungs, in the breast, or the colon. This model often reaches a failure point in advanced metastatic cancers. But once standard options are exhausted, the tumor frequently stops responding to treatments designed for its original location. A 2020 paper in Nature Cancer highlighted a frustrating paradox. Standard treatments like chemotherapy can actually accelerate this failure. When a tumor is hit with these therapies, the stress forces the tumor's genome to change. These forced mutations allow the cancer to adapt, developing drug resistance to survive the treatments meant to kill it. When cancer evolves this rapidly, treating it based on its anatomical starting point is no longer effective. Success now requires doctors to identify the specific shifting molecular glitch driving the tumor at that precise moment. To find that glitch, the Personalized Onco-Genomics program, or POG, sequences a patient's entire genome. The goal is to move past generalized treatments and find a unique vulnerability. This creates a staggering data challenge. The human genome contains roughly 20,000 genes, comprising 6 billion individual base pairs. Researchers use a comparative analysis to find the driver. They map the sequence of the patient's healthy cells side by side against the sequence of their biopsied cancer cells. The team scans for microscopic variations, the specific points where the cancer code has diverged from the healthy code. In this context, fighting advanced cancer shifts from a strictly biological procedure to a massive big data processing problem. Most of the mutations discovered in a tumor are passengers. These are dozens of harmless genetic changes that occur as a byproduct of cancer, creating a high volume of distracting data noise. The objective is to find the driver mutation, the single, lethal genetic alteration responsible for fueling the tumor's uncontrolled growth. Because the data is so complex, a human filter is required to find the signal. This is the role of the molecular tumor board. The board functions as a cross-disciplinary assembly, bringing together oncologists, geneticists, pathologists, and bioinformaticians to interpret the sequencing results in unison. This collaborative approach was used for Zuri Scrivens, a patient whose metastatic breast cancer had returned and was analyzed by the POG team. The sequencing revealed that her cancer wasn't following a standard breast cancer growth path. It was actually being driven by an insulin-like high growth factor. This discovery allowed the board to look for non-traditional solutions. They treated her breast cancer using a repurposed drug, commonly used to manage diabetes, which neutralized that specific growth factor. Pinpointing a tumor's driver mutation allows clinicians to look beyond anatomical categories. It provides a roadmap to select a specific molecule that targets the genetic glitch, regardless of where the cancer originated. This defines the new operating principle, treating a specific individual's mutation rather than an anatomical label. This logic explains why imatinib, a drug engineered for leukemia, is now used to treat stomach cancers. The two diseases appear different on an organ level, but if they share the same genetic alignment, the same molecule can stop them. However, this frontier has significant hurdles. Whole genome sequencing and molecular tumor boards are resource-intensive, expensive, and not yet available to every patient. There is also a biological time limit. A tumor's genome is never static. It will continually attempt to mutate around even the most targeted therapy. This means a treatment that successfully halts a cancer today may need to be completely replaced within a year as the genome adapts. Survival in this new era requires a continuous cycle of reading the code and updating the strategy. In the face of an evolving disease, a one-size-fits-all approach no longer applies.